Great. Welcome everyone to our Lunch and Learn talks. We have two presenters today, Mike and Jen, and I'll shortly introduce them after another. I just wanted to remind everyone that you're in listen-only mode, so if you have any questions, please write them in the chat so we can address them. After Mike talk is over, we're going to spend 5 minutes answering the questions for him, and then the same thing for Jen, and Katie's going to spearhead that and read out all the questions. So Mike is our first presenter. He is a current Knauss Fellow in the Ocean Acidification Program, and he's a PhD candidate at the Haskin Shellfish Research Laboratory at Rutgers University. His background is in shellfish ecology and sustainable aquaculture. Specifically, Mike's research is focused on developing the husbandry techniques for the Atlantic surfclam, breeding farm surfclam to be better adapted to ocean warming, and evaluating the feasibility of bivalve polyculture. And take it from here, Mike.
Thank you so much for that introduction, Anne. So I'd like to quickly introduce my co-authors, Dr. Ximing Guo and Dr. Daphne Monroe. And I'd like to just say a little bit more about my fellowship. I am the Knauss Fellow at the Ocean Acidification Program as their international and domestic liaison for capacity building. But today I'll be talking about my one of my dissertation chapters, which is focused on breeding farmed Atlantic surfclams for greater heat tolerance, the phenotypic and transcriptomic responses to heat stress. So as many people are aware, the oceans are warming, but the oceans aren't warming evenly across the entire world. In the Northeast, the oceans are warming faster than many other regions of the planet. In the Gulf of Maine, for example, warming is faster than 99% of global sea surface waters. And this is having impacts on numerous marine species. Shifts in species distributions are already evident. And one species where this is particularly well documented is the Atlantic surfclam. The Atlantic surfclam represents a lucrative and sustainable fishery in the northeastern United States. In 2018, 38.2 million pounds of surfclam meat was harvested at a value of $31.4 million US. As I mentioned, it's a sustainable fishery that's not overfished, and overfishing isn't occurring. And it also has very low bycatch. But one threat to the surfclam fishery is ocean warming, because surfclams are vulnerable to high temperature conditions that are going to be exacerbated by rising ocean temperatures. So surfclams are not only moving their range more northern to more northern latitudes, they're also moving into deeper water, which in effect is cooler water. So off the coast, deeper onto the continental shelf. Oops. Here we go. So in addition to surfclam fishing, surfclams are also a farmed species. Now, while surfclam aquaculture is still pretty new, it is expanding across much of the Northeast. In fact, NOAA has funded a number of surfclam aquaculture projects at our lab at Rutgers in southern New Jersey, but also in Massachusetts at Woods Hole. So these pictures here are showing what surfclam aquaculture looks like on some shallow New Jersey farms. And shallow is the key word here. All the aquaculture in New Jersey and along the East Coast is happening primarily in state waters that are relatively shallow, not the deep continental shelf water where surfclams are normally found. So right now on these farms, surfclams are experiencing temperatures that are right at the edge of their tolerance. So surfclams, maybe for a week or two during the year, are experiencing temperatures that are at their lethal limit. And this is only going to get worse in the decades to come. One interesting thing about surfclam vulnerability to heat stress is that their vulnerability differs throughout their developmental stage. So as larvae, surfclams are reach that lethal limit at about 25 degrees Celsius. And then once they metamorphose, their tolerance goes up a little bit. So juveniles hit that lethal limit at 26. Hit their lethal limit at 26 degrees. And once they metamorphose, the bigger a surfclam gets, the lower its thermal tolerance becomes. So a big adult surfclam that's caught in the wild fishery is at least 4 inches long. Their tolerance might be maybe 24 degrees. And then once you get to a monster 30-year-old surfclam that's 8 or 10 inches long, they can't withstand temperatures really above 20 to 23 degrees for very long. One of our earlier studies looked into the optimal rearing temperature for surfclam seed. So those are the juvenile stages between 1 millimeter and 10 millimeters. And what we found was that the lethal temperature was at 26 degrees, but this really varied across genetic cohort. So what this figure is showing on the y-axis is present survival, and on the x-axis is the different surfclam cohorts reared for 1 month after metamorphosis at these different temperatures. And what you'll see is that the survival really ranged quite drastically based on their cohort. So cohort B, for example, had a survival at 26 degrees, about 34%, whereas cohort C had survival at 26 degrees, about 15%. So that's a really big range to be observed for this species. So knowing that surfclams are going to be vulnerable to temperature stress currently on shallow coastal farms and in the future across much of their range, and knowing that there's this innate variability in how surfclams respond to temperature, we asked a series of questions related to the feasibility of breeding surfclams for greater heat tolerance. The first question was, how do surfclams respond to repeated exposures to lethal temperatures? Can selected surfclams be generated? Second question is, can surfclams be selectively bred for greater heat tolerance? And what that means is, do the progeny from those selected stocks perform better than the control clams? And then finally, what is the surfclam transcriptome under heat stress? What genes come on when surfclams are stressed? And does this gene expression differ across the selected stocks and across temperatures? And this might help with more sophisticated breeding techniques that might be available for bivalves broadly and surfclams specifically in the future. So the first experiment that we conducted actually used some of the clams that were inadvertently selected in that first experiment that I showed a couple slides back. So that experiment occurred in 2016 when we produced a surfclam cohort. And we had clams that were, to reiterate, selected at 26 degrees for 32 days. And then we had clams that were in control conditions for that period of time. So after that selection event or that experiment, the surfclams were reared for about 18 months. At that point, they became adult clams. They were sexually mature. And we assessed their heat tolerance by subjecting them to another lethal heat shock, either at 29 degrees for 12 days or to ambient conditions. So to show what that experiment looked like and to effectively test whether that inadvertent selection event was effective at producing a more tolerant stock of surfclams, we took the adult surfclams, crossed them across these two temperature treatments. Each one of those buckets represents a replicate, and within each bucket we had 6 individual clams. And this figure here is showing the survival of heat-selected and non-selected surfclams under heat stress. So on the y-axis, again, we have % survival, and on the x-axis, we have day. The solid lines represent the best fit model. The dotted lines represent the 95% confidence intervals, and each one of those dots represents the mean survival of the replicate buckets. And what we found was that both the day or the length of time in the exposure and also the progeny group, or the selected group in this case, were significant predictors of survival. So indeed, the surfclams that went through that early selection during their early juvenile phase were more tolerant to heat stress than clams that never experienced a heat shock before. So this was really exciting, and it basically allowed us to continue on with this process and see if we could potentially breed these animals for and develop a selectively bred line of surfclams. So can selected stocks be generated? The answer is yes. When juvenile surfclams were exposed to prolonged lethal temperatures, the adult survivors withstood subsequent heat stress for significantly longer than non-selected individuals. The next experiment we did took a new cohort of surfclams that was subjected to a selection event slightly differently. So this time we took adult clams and subjected them to 28 degrees for only 5 days. As I mentioned earlier, the bigger a clam gets, the less tolerant to heat it becomes. So it only took a little bit of time for 75% of the clams in the population to die. So what we were left with was these 25% that ostensibly were more heat tolerant. And then we also had a control group that came from the same cohort, but was not subjected to those conditions. So those clams were spawned in 2017. The selection occurred in the spring of, or the winter of 2019. And then in the spring of 2019, we back-crossed those groups. So the heat-selected clams were bred with more heat-selected clams, and the control clams were bred with the control clams. So the designation of their progeny are HSFF F119 and NSF119. So to kind of show some pictures of what that looks like, we have clams in the top left corner. Those are the adults, the brood stock. We use nail polish to designate the males and females. Don't worry, we didn't confuse the blue and the red from the control and the heat shock. We were meticulous at keeping the clams separate. And then on the top right, you see some buckets of surfclam larvae. On the bottom left, you see images of the surfclam larvae that were taken on a dynamic imaging device called FlowCam. That's me in the center holding some surfclam larvae caught on a screen. And then on the bottom right, you see a newly metamorphosed surfclam. So one of the things we wanted to assess was whether or not these two progeny groups of clams differed in their growth during their early development. So these figures are showing the growth of the two progeny groups as larvae and as early juveniles when they were reared using typical bivalve hatchery and nursery procedures under ambient conditions. So they weren't stressed, they were reared in the same exact conditions. How does their growth differ? And what we found was that their growth didn't differ. It didn't matter if you were a heat-selected or non-selected surfclam. When you were raised under these ambient conditions, your growth was exactly the same. So once the clams reach the size of about a dime, we call this seed clams, and this is the size that they would be deployed onto our partner farms. We decided to assess their heat tolerance. So we conducted a very similar experiment that was crossed. We took the heat-selected and the non-selected clams and exposed them to either a stressful condition, 29 degrees for 9 days, or a control condition, 11 degrees for 9 days. The only difference here was that we used 100 clams per replicate instead of 6 clams per replicate, and that was to keep the biomass the same in each one of the buckets. Obviously, it takes a lot more small clams to equal one large clam. So this figure is going to look familiar. It'll look virtually the same as the one a couple slides back. On the y-axis, we have percent survival. The x-axis, we have day. Each one of the dots represents the mean of the replicate buckets. Center line is the best fit model, and the 95% confidence intervals are represented by those dotted lines. And once again, what we found was that the exposure time and the progeny group were significant predictors of a surfclam survival during this stressful condition. And this was really exciting because what this means is that after just one generation of selection, we were able to produce surfclams that were more heat tolerant than non-selected surfclams. The biggest difference we observed was on day 6. So after 6 days at 29 degrees, the heat-selected surfclams had about 80% survival, whereas the non-selected surfclams, the controls, they only had about 50% survival. So this was a big difference, and I should note that surfclams aren't going to be experiencing 29-degree temperatures on farms currently, and that probably won't happen for very many years. But what we wanted to do here is kind of demonstrate this phenotype clearly in an experimental setting. Another thing you'll notice is that by day 7 and day 8, the two groups start looking a lot more the same, right? So after about 9 days, it doesn't matter if you were heat selected or non-selected, all the surf clams were dead. And effectively what this tells us is that additional rounds of selection are needed to really push this heat tolerant phenotype further and really differentiate between the heat selected and the non-selected surf clam groups. So to answer the second question, do progeny from selected stocks perform better than controls? Yes. Selected breeding enhanced heat tolerance in first generation surf clam progeny. Moreover, larval and juvenile growth of the heat selected progeny were not significantly different from that of control clams. And this was also an exciting finding because it means that there's no trade-off between growth and survival when the clams were exposed to stressful conditions. So moving right along to the third experiment that we conducted, and which was looking into the transcriptome of surf clams under heat stress. So for this experiment, we instead used the surf clam broodstock instead of the surf clam progeny. So the surf clam broodstock that was selected in 2019 were reserved after 3 months. The selection event occurred 3 months later. We took those clams and exposed them either an additional time to heat shock if they were the heat selected ones or control conditions. And then the naive clams, the controls, were exposed for a first time to stressful conditions or reserved in ambient conditions. And this exposure was sublethal, so only 6 hours at 29 degrees. Obviously, if we want to explore gene expression, we need the clams to be alive and expressing their genes. So we really wanted to see what does the immediate response look like in the surf clam during the stressful or shocking event. And just note a change in the initials. NS is equivalent to RC here. It just means random control, and you'll hear me going back and forth between non-selected and random control. It means the same thing here. So these are some images of what that experimental design looked like. Again, clams were placed into buckets in water that was heated or chilled. The clams were subjected to these conditions for 6 hours. And then the clams were removed. Their gill tissue was dissected and placed into a solution called RNA later. And then later, we sent the samples out to a biotech firm called Novagene and had the samples processed. Here are what our findings look like. So these images are showing the gene expression of heat-selected and non-selected or random control surf clams. On the figure on the on the left-hand side, you're seeing a cluster analysis. So each one of those slices is a different gene, and the colors represent their gene expression. So red is something that's very highly expressed, and the deeper the red, the more expression there is. White is kind of moderate expression, and then deep blue is showing down regulation or a lot less expression than you would otherwise expect. And on the right-hand side, you have those Venn diagrams. What that is showing is the number of differentially expressed genes across group and temperature. So on the top, you're having the within group across temperature comparisons. And on the bottom, you're having the across group within temperature comparisons. So the first one we'll start with is the surf clams that were exposed to 16 degrees. So only 31 genes were differentially expressed between the two groups when they're exposed to 16 degrees. And I'll use the cursor here to point out which two groups I'm talking about. You'll see that largely these two columns look identical. So it didn't matter that this group was had undergone that selection event. When they're in control conditions, they're kind of up and down regulating the same set of genes. Now, if we look at the two groups when they were exposed to 29 degrees, 51 genes were differentially expressed. And you'll see that, yes, largely the expression patterns look different. There are these big chunks of underexpressed genes and these three and these big chunks of overexpressed genes. But the one difference is the level of expression. The naive clams actually had much more expression for more genes. They had a really robust response to this heat shock. And remember, this was their first experience in this condition. Next, if we look at the comparison between the control groups when they were at at 16 degrees or at 29 degrees, you'll see that they upregulated over 4,000 genes. And if this column, RC29, looks radically different from RC16. And that is similarly present when we compare the heat-selected clams at 16 versus 29 degrees, but about a third less genes were differentially expressed. So again, they look pretty different, but the level of gene expression was not nearly as robust as the control clams. And this figure here is showing some of the notably differentially expressed genes. So I'll first focus on the clams when they were exposed to 29 degrees, and you'll see that the naive clams were upregulating lipid biosynthesis and homeostasis genes. And what our hypothesis is that one of the effects of heat stress on surfclams is that their membranes start to break down. In hot conditions, their membranes become too fluid and they start becoming leaky. So it's possible that the RC clams, or the control clams, were modifying their membranes to cope with this heat stress. So they're building up different types of fatty acids that are more robust against that heat stress. On the flip side, the heat-selected clams seem to have a more robust stress response. So they're upregulating the heat shock protein 70, which is commonly seen in stressful conditions, particularly heat, immune response genes, apoptosis genes, DNA regulation. So it's possible that these clams, because they had already been selected to withstand this temperature, potentially already remodeled their membranes, so they didn't need to upregulate those genes, but they were better adapted at upregulating these stress response genes. And then if we compare on the flip side, the two groups when they're at ambient conditions, again, only 31 genes were differentially expressed. You see some differences in cell signaling, protein binding, and apoptosis, meaning that there are probably some differences between these two groups, even when the clams are not stressed. So to answer this third question, how does gene expression differ across the selected stocks and across temperatures? Heat shock induced a significant response in both the selected and non-selected broodstock groups. However, the naive clams exhibited a more robust response than did the clams that survived a previous heat shock. So what one of our hypotheses is that potentially, because the first shock and the second shock was only about 3 months apart for the HS group, it's possible that the gene expression levels were lower because a lot of the gene products were still residing in their cells. So they already had that massive upregulation of genes. The proteins that they created were still lingering in their cells, so that when the second shock came, they didn't need to expend energy creating all those gene products. They already had the protective proteins present. Again, this is all our hypotheses, so this definitely is going to require more research. So to conclude, the results from these experiments suggest that heat-induced selection can identify clams genetically predisposed to withstanding high temperature conditions. And in 2020 and 2021, additional rounds of selection will occur. And we will continue to assess the performance of progeny groups in the field on our partner shellfish farms. I didn't talk too much about that, but that is rapidly occurring over the next couple months. So finally, I'd like to acknowledge a lot of members of the Haskin Shellfish Research Lab and the New Jersey Aquaculture Innovation Center who definitely played a big role in helping this research be conducted, as well as our partner farmers and our funding and support from NOAA and New Jersey Sea Grant, USDA, NESARE, and Seabird Scientific. And with that, I'll take any questions. Thank you very much.
Thank you so much, Mike. We do have one question so far from Vince Gita. What do different cohorts represent? Different sources, different spawnings, or what?
Different cohorts mean different genetically, so different spawns and different genetics. So to kind of say it a different way, so in experiment 2, for example, we started out with a single cohort of clams that was that constituted multiple spawns. And then those clams were then divided in half. So they had the same parentage. They were divided in half. Part of them was subjected to the heat shock to produce the heat-selected clams, and part of them were reserved as those control clams.
Okay, thank you. We do have one more question. At high temperatures, everything dies in a matter of days. Can you select clams that will survive long enough to grow to market size?
Yes, so this was something that I forgot to mention. Clams in the wild fishery are harvested as kind of older adults. So they're at least 6 or 7 years old. They have to be at least 120 millimeters or larger than 4 inches. So those are kind of the big clams that are caught for industrial fishing purposes. On farms, surfclam aquaculture, the clams are raised only to about 2 inches long. So think more like a little neck size or a pasta neck, as some people like to call the little surf clams that are great with linguine. Now, those clams actually could reach market size in about 15 to 18 months. So they really only have to experience one summer on the farm where they might be exposed to those stressful conditions. So today, surf clams can be raised on shallow farms across the Northeast. And even though the temperatures might get a little warm, surf clams could really cope with it. Our hope is that we can produce surf clams that will be able to withstand some of those high temperature conditions for longer. Although eventually when you get to something like 29 or 30 degrees, it's probably not possible to produce a surf clam that is that tolerant to heat shock for more than probably, like 9 or maybe 15 days.
Great, it looks like we have 2 more questions.
Sure.
Have you considered how transgenerational plasticity, the ability of progeny to alter their phenotype without a change in genotype, might synergize or interfere with selective breeding programs?
Yeah, that's something that we've been thinking about a lot at our lab, is kind of the role of plasticity and also whether epigenetics is going to be playing a role in kind of whether or not surf clams could be adapted to heat tolerance very quickly. Across other shellfish species, we see kind of how surf clams or bivalves can be selected relatively quickly just due to husbandry techniques for various kind of, in effect, become domesticated rather quickly. So it's something that we're thinking about often, but I don't have any kind of specific comments to make about those effects in this study. It's something that we're thinking about, and we think that the data here show pretty clearly that something is genetic and not epigenetic based on the way these experiments were conducted. But it's something that we're thinking about, and when we design experiments in the future, are taking into consideration.
Aside from yours, have there been any experiments or studies looking at the heritability of this trait, heat tolerance, and the potential for it to be incorporated in a breeding program?
To my knowledge, no one else is doing that for surf clams, but I believe there are other species, other bivalves, where that is an active area of research. The first one that comes to mind is looking at Manila clam breeding in China. They've seen a lot of really devastating summer mortalities in their Manila clam farms. So that's an active area of research on that hemisphere. But to my knowledge, Rutgers is the only group looking at selectively breeding surf clams for heat tolerance.
Okay, thank you. And the last question is, are you planning to look at other factors that influence clam growth like pH, synergistic response to heat, stress, and pH?
That wasn't something I was originally planning on studying, but now that I've joined the ocean acidification program, it's something I think about all the time. And I think in the next, I think OA, there's some other research out there that has shown that surf clams are pretty vulnerable to acidic conditions. So I'd be really interested in seeing how those two variables interact to affect surf clam survival. I'm on the tail end of my dissertation, so I probably won't be the one to do that work, at least as a PhD candidate. But it's definitely something I'm very interested in, and other people will likely investigate.
Okay, thank you, Mike.